Alrighty. Good morning, everybody. How was everybody's weekend? Not bad. Cool. I'm gonna pass back your exams. I don't know if you saw already, but I did post the grades on Blackboard. So it was the first exam, so I wasn't sure, how it was gonna go for you guys. I was hoping for an average of about like a C plus to B minus, and the average was indeed a 77.6. So we like hit that right on the nail. But there will be a curve towards the end of this semester. So instead of like making like a curve for each exam, I will have some sort of curve where you guys can get back points towards the end of the semester. So just keep working hard, keep trying, keep studying, keep asking questions, and you should be fine. If you do have questions about any of it, you're welcome to stop by my office and we can talk. So I do have your exams with your Scantrons attached, so I'll pass those back. Yeah. Jesse. Page. Oh, so Lucia. Last. Nicole. Yeah. Monica. Autumn. Tyler, she's on here as well. Tegan. Erica. Abby. Brooke. Ryan. James. Victoria. All right, so for the exam, well, you guys could like take a look at it for a minute, but then let's put it away and move on to the next lecture. And if you guys have any questions about the exam, I just ask that you guys wait 24 hours and actually like look through and make sure that if you want to come to me and like, say like, oh, I think that whatever your answer key is wrong or whatever, like give it 24 hours and then come to me. All right. All right, so can you guys put your exams over? All right, cool. So today we're gonna start a second unit. We're gonna start off with lecture 6, and we're gonna be talking about some viruses and phage. The majority of this class will be on bacteria. This will be one of the few lectures on viruses and phage. It is very important to know, things about viruses and microbiology, so we'll go through some of the important things in this lecture. So some of the suggested readings, just the first 4 sections of chapter 6. And as a little overview, today we're going to be talking about what a virus is. We're also going to be talking specifically about viruses that infect bacteria, which are called bacteriophage. We're going to talk about the genomes of viruses and how they are structured. And so here, for example, I'm showing you on the left this coronavirus. So it has these infamous spiked proteins. That's why it gets the name corona. The spiked proteins are like crown. That's what they mean. Corona means crown. And so, yeah, and so it has this membrane-bound nucleocapsid, and this will make a little bit more sense a little bit later in the lecture. And then over here on the right, I'm showing you this more common example of what a bacteriophage looks like, these phage that infect bacteria, basically. And so they have this capsid head, this tail sheath, and then these tail fibers. It kind of looks like, I don't know, some alien coming in with legs to infect. And then we're going to talk a little bit more about the 3 life cycles of bacteria, the bacteriophage in particular. So just some nomenclature for you guys. So viruses are considered a microbe, but they are not alive, so they can't reproduce autonomously, and so that's the reason why we don't consider viruses living things. But what it can do is it can exploit the cellular machinery of a host in order to replicate itself. So it subverts the cellular machinery of the host in order to produce viral particles. So that's how they are able to live and maintain, their reproduction. And so there are viruses that infect specific cell types, and all cells have some virus that can infect them. So all cell types can be infected by a virus, but specific viruses have a certain host range. So each virus has a very specific host that it can infect, but all cell types are able to be infected by viruses, and I'll talk a little bit more about why there's that potential. One virus particle is called a viron or viron, and so that will consist of one virus particle will consist of some sort of single nucleic acid, so it'll be either DNA or RNA, and it'll be protected within a protein capsid. And so, like I said, different viruses infect different hosts, and usually the viruses are limited to a particular host range of either closely related strains or species. So there are some viruses that are specific to infecting E. coli, for example, and we'll talk a little bit more about those. So each virus particle has to have some sort of nucleic acid, DNA or RNA, and it's protected within some sort of protein capsid. And so before we go in depth on bacterial viruses or these bacteriophage, I want to talk a little bit about eukaryotic viruses and their taxonomic features. And so here in green, in this green box, what we're looking at is these eukaryotic viruses that have DNA as their genome. In orange, we're looking at viruses that have RNA as their genome. So like I said, their genome can be either DNA or RNA. And so here in the a little bit darker orange, you see that single-stranded RNA. There's very, very few viruses that make double-stranded RNA. If it's double-stranded, usually it's DNA, but there are exceptions to the rule, so that's why this is a smaller box. But just it's important to know that there are some that have DNA or RNA as their nucleic acid type. And if you look specifically at the single-stranded RNA, in the top box you have this minus, this negative, and then in bottom box you have this plus. And so this plus or minus stands for the RNA being positive or negative sense. And I'll explain what that means. So a positive sense virus, like coronavirus, that's right here, means that the viral RNA is the same sense as the mRNA. And so what that means is that when the viral RNA gets integrated into the host cell that it's trying to infect, it can immediately be translated by the host cell machinery because it's in the same sense, it's in the positive sense as the RNA of the host cell that it's infecting. If it is negative sense RNA is complementary to the mRNA sequence of that host cell. Yeah.
So Does it like only relate to the effectiveness, or does it also relate to its range? Like it can only infect the positive or negative cells?
So it's not it's not the host that's positive or negative sense. It's the virus that's positive or negative sense. So the virus will either have RNA that's positive sense, which means that it's mRNA, or RNA is the same sense as the mRNA in the cell that it's infecting. Does that make sense? So if it's negative sense, it can still be translated, but it first has to be converted to a positive sense. So it doesn't like make it more specific or anything in that sense. It's just, it is like what it is. Some are positive, some are negative sense. If the virus is in the positive sense, it's gonna have an RNA that's complementary to the host mRNA that it's infecting, and it could just immediately be transcribed. Whereas if it's negative sense, it just needs to be converted into positive sense for it to be able to utilize the host cell machinery and be transcribed. Does that make sense?
So there's one step extra for the negative sense to get translated.
Yeah, there's one step extra, exactly. You just have to know that if it's negative sense, the RNA is negative sense, which means it's complementary to the mRNA sequence. So it's got to be, you know, it's got to be converted into the other sequence in order to be translated. And as we go through this a little bit more, hopefully it'll click a little bit better. Okay, and so, yeah, and so the last thing here is that there are double-stranded or single-stranded DNA or RNA, and like I said, double-stranded RNA is like really rare, but it can be in either form. So the virus itself can have a membrane around it, and those viruses are called enveloped. But if you have just the protein capsid, then it's non-enveloped, or it's naked. So all viruses will have their genome, their DNA or RNA, and they'll have the protein capsid. Some of them will have a membrane that encloses it or envelopes it as well. Not all of them do. So if they just have the capsid, they're considered naked or non-enveloped. So capsid shape, so there's some varying shapes of this like capsid head. They could be filamentous, they can be icosahedral. So over here, we're looking at a filamentous one. We've talked a little bit about filamentous organisms like fungi and bacteria, and so viruses can also be filamentous, which is they have these long filament-like structures. They can also be icosahedral, which is this, you know, icosahedral shape that's very common in schematics of viruses. And then there could be other more sophisticated shapes that we're not going to talk too much about. And so just something else to know is that eukaryotic viruses can assemble in either the nucleus or the cytoplasm of the host. So you don't have to know all of these taxonomic features and structures of viruses. The only one that I think that I would want you guys to know is this coronavirus. So let's do a little concept check. I guess it's just good to know since it's very relevant to all our lives at the moment. So coronaviruses have which of the following features? So we have our DNA and our protein capsid, and we have this like envelope outside of that protein capsid, and then you have these like corona spikes. So first, is it enveloped or is it naked? Okay, so we'll cross out B and D. And so is it positive sense, single-stranded RNA, or negative sense, single-stranded DNA? Yeah, so A. So corona is an enveloped, single-stranded RNA virus in the positive sense. All right, so moving on from eukaryotic viruses, we are going to talk a little bit more about bacteriophage. So viruses can be classified by the types of hosts that they infect, and so as the name suggests, Bacteriophage is a virus that infects bacteria. They also are called bacterial eaters. And we study them in the lab by plating on a lawn of bacteria. So over here, what I'm showing you is this petri dish, and all of that, not the circles, the actual plate has bacterial growth on it, and we call that a lawn of bacteria when you have just a lot of bacterial growth. And so here, what we're looking at is these plaques. So basically, these plaques are formed. What this person had done here is they grew up all of this bacteria, and they put phage that eats this bacteria on the plate. And so the phage, one virus particle, will start to eat away at the bacteria. And so when it does that, it's able to replicate because it's using the cell machinery of the bacteria to replicate itself, and it's producing more and more and more phage. And so all of those progeny phage are now eating away at the bacteria that are in that zone. And so that creates this zone of inhibition, which means this plaque, that's where the virus has eaten the bacteria. So it's kind of different. It's kind of the opposite concept of seeing the colonies on a plate from bacterial growth, where you actually see the visible colonies. This is kind of like the opposite, where instead of seeing colonies, you're seeing bacterial death, and that's shown or displayed by these plaques. And so if you pick a plaque, in the very center of that plaque is where there was one single phage, one virion particle that infected bacteria. But because they can replicate so fast and they use the bacterial cell machinery to do that, that's why you have this bigger circle, and that's because of all of the progeny phage that have begun to eat away at the bacteria in that spot. Does that make sense? So this is an example of how we're able to study bacteriophage by growing them on a plate. It's possible that we will do an experiment like this in the lab. I just have to make sure we have all of the things that we need for it. But yeah, it's pretty cool to see. Many bacteriophage are tailed phage, so that's this example right here. You see that they have these tail fibers, and we'll talk a little bit more about that. But I guess just to give you a little bit more perspective, their genomes can vary, so their DNA or their RNA can vary. Some can be circular, most are linear. Like I said, they're single or double-stranded. But to give you a little bit of perspective, phage are incredibly diverse. So when I say phage, I'm essentially referring to bacteriophage, just so you know the terminology. So phage are incredibly diverse, and they're very abundant compared to living things. So to give you some context of just how abundant phage are on Earth to other living things, the number of human cells that we each make is about 37 trillion. We also make about the same number of bacteria, and I've said this before, we are essentially half microbial. And so the number of bacteria on Earth is ridiculously higher than that. And so if you were to count all of the bacteria on Earth plus every living thing on Earth, that equals to not even close to the number of phage on Earth. So the number of phage on Earth is 10 to the 31. So that's a 1 with 31 zeros. That's a huge number. So another little spiel. There are 1 billion more phage than stars in the universe, 10 billion more phage than grains of sand on Earth, and 250 million phage in 1 drop of seawater. So there's so much phage. So with so many phage on Earth, should we humans be worried? The short answer here is no. And that's because phage only have mechanisms to infect bacteria. There is no direct harm to human cells whatsoever, but the key there is that there's no direct harm. However, because they infect bacteria, and since we are half microbial, of course, phage will have indirect effects on human health. So phage are very host specific, and like I've said already, a specific phage can infect usually a certain species of bacteria. And the specificity comes from recognizing receptors that are exclusive to the surfaces of bacterial cells. So over here, I'm showing you phage, and there are this red, yellow, and blue receptors. And so these receptors are on bacteria that the phage can recognize. So if the phage recognizes the red one, it's gonna go in and attack the bacteria with the red one. Yeah.
So is this where you can get bacterial and viral infections at the same exact time? Like when you're more predisposed to one, it tends to, like you can also get a viral infection at the same time because of bacteriophages?
That's a good question. I don't know that I can say for sure that that's why from a clinical perspective. I know that, you know, with viral and bacterial pneumonia, for instance, is kind of like where my brain goes with that. I don't really think that it has to do with the virus, like the viral pneumonia is infecting the bacterial pneumonia, because these viruses just eat like bacteria. So it doesn't have to do with them infecting humans. So indirectly, you can have effects, and we'll talk a little bit more about that. But no, I think that in that case, having a viral infection is a direct effect onto the host. So that's not the bacteriophage, if that makes sense. But that's a really good question. Yeah. But yeah, so basically the phage are performing this bacterial genocide every single day. They're constantly attacking microbes and trying to keep them in line in our immune system. And they just have a lot of, you know, positive and negative effects in that case. So the structure of phage here, so the phage structure, basically they all have this capsid in some form, and it keeps its genome intact, and it also enables infection of the right host cell. So we have these tail fibers that recognize specific surface structures on bacteria. The capsid packages and protects the phage genome from degradation. So usually the DNA or the RNA is inside of this capsid head. And so it protects it specifically from degradation. So if you have DNA out in the environment, it can easily get chopped up by DNases. So this is the protein shell, the capsid, that protects the DNA or RNA from degradation. And so phage can have different forms. So you can have these tailed phage. So if they have these tail fibers, that's when we call it tailed. And then it can also be icosahedral. So that just means it's tailless. So it has just the plasmid head without the tail. And like I've mentioned, they could be filamentous, like this M13 virus here. You don't need to know specific names here, but just know that they can have different forms and what those forms look like. Any questions? So filamentous viruses are a major category of virus structure. The most famous one is Ebola. And so this is a filamentous bacteriophage that infects E. coli. Or sorry, M13 is the one that infects E. coli. And so this one in particular is the example that we'll kind of be using to talk about filamentous phage. But essentially, the capsid consists of a long tube of protein with the genome coiled inside. So it's a relatively helical, simple helical capsid that surrounds the single-stranded DNA genome that's coiled within it. And so with these M13 phage, the length of the genome dictates the length of the virus. So if they vary in length, and it depends on the genome size. So if the genome was like halfway over here, can you guys see my cursor? No? Yes, maybe. Yeah, so if the genome were to end like right here, then the capsid would close off right here as well. So it depends on how long the genome is, that's how long the actual virus is, the capsid. And then notice that at the end we have these like tail fibers, and so these mediate specific attachment to the pilus of the host bacteria that they like to infect. So E. coli has these pili, and so this M13 recognizes E. coli through these fibers at the end. Any questions here? Okay. So tailed phages are pretty cool. I think I like them a little bit, liked learning about them. So some phage supplement their icosahedral capsid with an elaborate delivery device. So this capsid head has this really elaborate delivery device that we call the tail sheath. And so this tail sheath right here is kind of like the body of it. And so that is used as a delivery device. And then these tail phages also have these tail fibers that allow the phage to stabilize itself onto the host bacteria. So what I'm showing you here is T4 bacteriophages. So this is the common structure of a T4 phage. These also infect E. coli. It's one of the main ones that are studied. And you have this head, which is the protein capsid that contains the double-stranded DNA genome in this case. But this genome is really pressure-packed. So there's a lot of DNA, but it's really pressure-packed in there. It's important to know that, and I'll explain why later. This tail sheath kind of has this covered neck collar, and so this is the delivery device. This is what channels the genome into the cell. And then we have these tail fibers that help stabilize the phage onto the host cell surface. So over here, I'm showing you basically an E. coli cell, and these are a lot of phage that are infecting it. So over here, we have the head of the phage, and then we have the sheath right here. And then unfortunately, we can't see the actual tail fibers because the tail fibers are the ones that will actually connect to the host cell and then allows this to kind of sit down and like be able to inject the DNA in here. And so as you can see here, there's one of them. Like if they're white in the center, that means the DNA is still in there. If they are clear, we call those ghost cells, and that means that the DNA has been inserted into the cell. So that's one of the ways that we've been able to study them with this EM imaging. So a little concept check. What do all viruses contain? Sorry? D. So are all viruses enveloped? C, exactly. So A, B, and D are incorrect because not all viruses contain an envelope. Remember, some of them are naked or non-enveloped is another way we refer to them. But all viruses contain a genome and that protein capsid to protect the genome. So we know most about bacteriophages that are found in our gut, and obviously if there's an association with human health, we know more about the biology of those phage. So so the best known bacteriophages are those of the mammalian intestinal tract. Gut bacteriophages are sometimes called coliphages. So remember when I told you E. coli, the coli stands for colon because that's where E. coli is mostly isolated from. This is another instance where we're seeing this. So coli means in the colon or in that GI tract area, so they can be called coliphages. And they're important because they're part of a microbial community that can actually help modulate our immune system. So eventually I'll show you how they can have the ability to indirectly affect our microbiome, and as a result can modulate our digestion, immune system, and mental health indirectly. And so phage can control the diversity of the microbes in our gut in order to modulate our own health. So they can help to limit bacterial levels in our gut to help our immune system, remain tolerant to things. So this is one way that they can be helpful, and they can also, attack biofilms. They can attack, destroy, and dissolve biofilms if they feel like there's like a population happening, and it's a host for them, and so they're indirectly helping us. So they can be good too. Any questions on this? So let's talk a little bit about how infection occurs. So the first thing that has to happen for a phage to infect a bacteria is it has to recognize its host. So to start an infection, the phage has to contact and attach to the surface of the right host cell. And this contact and attachment are mediated by cell surface receptors. So like I said, there are specific cell surface receptors on the bacteria that are there that the phage can recognize. And so one thing that's important to note is the bacterial cell receptors that the phage target usually are those that are really important for the bacteria to make. They're usually essential for the bacteria. And so because of that, it doesn't allow the bacteria to evolutionarily delete that gene to evade infection. So if bacteria makes a protein on its surface that phage normally recognizes, and that protein isn't really important for its survival, and the phage keeps on eating and killing those bacteria, then you would think evolutionarily the bacteria are able to mutate so that the phage can't keep infecting. But the phage are, I guess, evolutionarily smart in a way where they tend to pick bacteria as a host with specific receptors that the bacteria needs to make so that it can't just delete it and not be a host for the virus. Does that make sense? So here is just an example of ways in which phage can recognize Salmonella and Enterica as the correct host. So it's really not important for you guys to remember that it's Salmonella that it's infecting, but there are various molecules that can serve as these phage receptors. So we talked about the LPS components on cells. So you're on a gram-negative cell, you have this LPS that could be something that the phage could recognize. You could have outer membrane proteins that they can recognize, transport channels, or flagella and pili, like I mentioned in the E. coli example. The M13 recognizes the flagella or the pili on E. coli. So binding is highly specific, and all of these can be used as components that the phage recognizes its host bacteria. So after attachment, so after the phage attaches, the bacteriophage injects its genome. So how does it do this? So it's important to mention here that it's these tail fibers that are attaching to the cell surface. And so basically what happens is it attaches to the cell envelope, and then you have this sheath. This sheath has to literally squat down and contract itself and brings the head near the cell surface in order for it to inject its DNA. So the sheath has to literally squat down and poop out the DNA in this case. It's like the best way to put it, but it helps you remember it. And so remember I said that this DNA is extremely compact. So basically there's a reason for that. And so there's this pressure of this spooled DNA that when this sheath is contracting and it opens its site to release the DNA, there's all this pressure that's gonna force the DNA to shoot out because it's so compact. And so here in this example, I kind of alluded to this a little bit. You can see that this phage capsid now remains outside and attached to the cell in this case, but there's no DNA here, so it's gonna look like a ghost phage, whereas this one still hasn't injected its DNA. So only the DNA gets injected into the cell, and the tail fibers are what bind to the cell envelope in order to inject its DNA. The capsid remains outside, the sheath also remains outside, but the sheath does contract to bring the head closer down. Does that make sense? Okay. Let's make sure I didn't miss anything there. Yeah. So I'm just going to show you a little video reiterating this. So this video is illustrating what I just told you. Here's a T4 bacteriophage, and it's looking for a pore victim. And so its tail fibers will find cell surface receptors, just like that. And after receptor recognition, the phage will squat down. The base plate opens. That will then allow the DNA to be injected. So here it's squatting down. This base plate is being opened and it's injecting the DNA. And then there's injection of this genome. Into the cytoplasm. So bacteriophages, they infect a host cell. So now the viral genome is in the bacteria. So there's different things that have to happen now. So we talked about how it gets its DNA into the bacterial cell, but now what happens? And so it actually has to make a choice. So some viruses, so I said that viruses like to replicate, and they do, but some of them do more than just replicate. Some of them just stabilize their genomes into the host. So we call those prophage, when the viral genome is stabilized into the host. And so what that means is, once the virus injects its DNA over here, it can either be its own circular chromosome, which we call a plasmid, or it can actually integrate the DNA into the DNA in the cell as well and kind of hide out and just become a part of it. And so, but whether it does this or this, we call this a prophage when the DNA is of the phage is also inside the cell. Yeah.
Have we been able to like sequence any bacteriophages?
Oh yeah. I haven't done any of it, but we have sequencing done for sure. Yeah. That's a good question. Yeah. Any other questions there? Okay. So the viral genome is now in the bacteria, and it either circularizes or it can integrate its DNA into the actual genome of the bacterial cell. And so basically, after this, it has to make a choice, and the phage can undergo one of 3 cycles. So we're going to talk more in depth about these 3 cycles, and then we're going to end lecture because viruses and phage are scary, and I don't want to get into too much depth, and I just, to teach, it's just like, more difficult. And so I'm more into bacteria and fungi, and so I think these are the really important things to know. So we'll go through these. So in the lytic cycle, as soon as the DNA gets in, it exploits the cell machinery of the bacteria, and then it's able to make more progeny, phage, so basically it replicates. It busts open the bacterial cell, destroys all the cells around it, it kills it, and it just continues to make more phage. So in the lytic cycle, so lytic comes from, like bacteriolytic, I guess is one term that you may have heard before, which just means death. So they like to kill. In the lysogenic cycle, it inserts its DNA and it stabilizes its viral genome in the host cell. So it becomes this prophage where it just stabilizes it in the host. And so basically it's like a sleeper cell. So the virus realizes that this cell is kind of like a mansion for it with all these nutrients. So it just wants to stick around. The virus doesn't have to do any of the work to replicate itself because because the actual cell is doing the replication. The cell can still replicate itself, but as the virus is kind of hanging out there, the virus gets replicated too. So it's just taking advantage in that way. In this case, in this lysogenic cycle, this is where you can have a switch to the lytic cycle. And so basically, if the bacteria gets into an environment that's not favorable, where nutrients are scarce, or there's DNA damage out there by UV light, then the phage has to get out. It realizes something is up and I don't want to stay in this mansion anymore. I have to get out, so it can then bust open this cell and switch to the lytic cycle. So it kind of can hang out as long as it needs to until things don't become favorable for the bacterial cell anymore, and the phage realizes, I gotta get out of here. And so the slow release cycle is the third one that's a little bit more recently studied. And so this one is considered to be a persistent phage infection. And so the reason being is the cell, the bacterial cell that the virus is infecting, is kept just barely alive. So it's kind of like a zombie cell. So basically, the cell is continuously spewing out phage, but the phage doesn't kill the bacteria. It just keeps this factory alive for the virus. And so the bacterial cell is still able to grow, but it's more slowly than uninfected cells. So that's me kind of talking at you about these, but I also have details. So in the lytic replication cycle, it all starts with host recognition and attachment. And so basically it has to adhere to the host cell. And then, so here we're looking at an example where you have the phage particle out here. This is the bacterial cell, this is the bacterial DNA in the light blue. And so the phage DNA in purple gets in, and then it naturally will circularize in there. And then because now it's in there, it can exploit the cell's machinery in order to make proteins and things for itself. So basically, the genetic information here is used to synthesize the proteins that are necessary for the progeny phage to be forming. So as these progeny phage are being replicated, so now we have progeny phage being replicated, we also have the viral DNA being replicated as well. So once you have enough of all of these, the viral genome begins to get packaged into these prophage, or these progeny phage. And so this gets packaged in there, so as you can see here, it gets filled up with the purple DNA. And so then once there's enough viral progeny assembled, then the cell can lyse, the phage bursts open the cell, kills the bacteria. And then once the progeny phages are released, if there are any neighboring bacterial cells, those cells will get infected too, and it does it over and over again where it kills, kills, and kills. Any questions on this cycle? Okay, so one thing I want to kind of reiterate, because this was a concept that I had to read through a little bit more, is here we're seeing that the viral DNA is inserted and it circularizes, and so normally this is going to start replicating itself, but there are all these little proteins here, these capsid proteins that are being assembled because we have this viral DNA here. It's going to start utilizing the cell machinery to pump out these capsid proteins, which then assemble into the actual capsid and the sheath, and then the DNA can get in there, and that's why you have those kind of popping up out of nowhere, if that makes sense. Okay. So the lysogenic cycle is this temperate phage, kind of this alternative pathway for the phage to be able to maintain their genome as a prophage in the cell. So prophage is when it stabilizes its DNA in the cell, kind of becomes a sleeper cell, so it exists happily. So these temperate phage infect and lyse cells like a lytic phage, but they also have an alternate pathway where it can maintain its genome as a prophage. So we call temperate phage the phage that are able to switch between lysogenic and lytic. So that's that terminology there. And so the phage lysogenizes the host, so it leads to this state called lysogeny. So the prophage is, like I said, a sleeper cell that remains inactive. It doesn't have to do anything on its own to replicate because it uses the bacteria's replication of itself to just hide out and be replicated with it. So the phage are quiescent as a prophage when it's in this lysogeny state. So that's important for you guys to know. And so the advantage here is that the prophage, the stabilized phage, inside of the cell is replicated and inherited along with the host chromosome each generation that it divides and replicates. And like I said, it can become lytic and abandon ship when there are environmental stressors for the bacteria, because we talked about bacteria a lot and how they kind of go from feast to famine a lot. So they're not going to be just feasting and happy all the time. And so this is why there's these temperate phage that essentially have to switch from lysogenic to lytic in order to be able to combat these environmental pressures. And some of the things that happen to the bacterial cell that will cause the virus to want to abandon ship and switch to this lytic cycle is UV exposure that basically damages DNA, nutrient deprivation, and there's a number of other triggers. So when the bacterial cell is threatened, this can trigger the phage's switch to the lytic cycle. So phage lambda is a very well-studied phage that infects E. coli. It's the classic example of this temperate phage. So just to go through this a little bit, you have the host genome. The phage attaches to the host cell, it inserts its DNA. So this host attachment and insertion of the genome is similar, and then the DNA will circularize. We went through what happens in the lytic cycle. So in the lysogenic cycle, this phage DNA, the purple, integrates into the host genome to form a prophage. So this is a prophage now. And so now this integrated phage DNA reproduces with the host genome. So now we have replication, and so you have bacterial cell replicating. Remember, the phage isn't doing anything itself. But for instance, if there is stress that's induced for the bacterial cell, this induces excision of the phage DNA where it gets excised out, and then the phage recombines by joining its ends again by forming the circular plasmid, and then it enters the lytic cycle again. Does that make sense? So, yeah, it's important to know that this phage can exist as integrated into the host DNA, or it can be its own plasmid still and do this, but regardless, if it's just hanging out, it's called this prophage. And so this decision between the two cycles is dictated by environmental cues. So events that threaten the host cell survival will trigger a lytic burst. So I'm just kind of reinforcing some concepts here just so that you guys start to get familiar with more of the terminology. Okay, and so lastly, there's this newly identified life cycle for phage called the slow release cycle of a persistent phage. So this is what I like to call these zombie phage. So basically, there are these zombie phage replication factories where the host continues to reproduce, but more slowly than regular cells that are uninfected. So this is an example of a filamentous phage. That's just the example here. It's not necessarily just filamentous phage that do this. But basically what it does is it infects, so it inserts its DNA in there, circularizes, and so at this point, this DNA is replicated, so that's why you have this replicative form where you have this kind of just replicated. It has to do that in this slow release cycle, so it forms this double-stranded circle. And so now it's basically DNA is and so basically, yeah, the DNA is replicated into these single-stranded DNA, and these phages can start to assemble, and you start to get more and more of this phage DNA, and they exit the cell, but without lysing the cell. So that's the key here, that the cells here, the bacterial cells, are still able to do their thing and replicate themselves, but the phage is also able to utilize the machinery of the cell and its replication in order to replicate itself and, you know, get pumped out of there. It also utilizes the cell's machinery in order to make these proteins the same way that it did in the lytic cycle, but it's, and so basically it can then, you know, get these proteins around the DNA and create that capsid and then get released. But the bacterial cell remains intact, it doesn't lyse. So it's important to realize, to note here that the host continues to reproduce similarly to the lysogenic cycle, but it's different because it reproduces more slowly than uninfected cells, because the cell machinery is actually being used to make these phage and to make these protein capsids. So the resources are being used to make phage, so that's why this is a much slower cycle for the bacteria. So this is the third way that phage can persist. So this is, you know, one of those that has some combination of lytic and lysogenic, but it's its own thing. So make sure you keep that straight. Any questions on that? Okay. So shorter lecture, but a lot of content, a lot of new content, and I kind of threw everything at you that you should know for phage in this lecture. We might go through a little bit more of viruses next lecture, we'll see. But yeah, let me know if you have questions. Recording will be on Blackboard, and I will see you guys tomorrow.
I want to go ask him about